Questions about gram-positive cocci. Question 1. True statements about staphylococcus infections. 1. Sulfadiazine and penicillin are effective for treatments. 2. Staphylococcus infections causes operative virulent inflammation. 3. Erythromycin is effective. 5. It produces enterotoxin. Explanation. Staphylococcus commonly causes purulent infections due to strong inflammatory responses in tissues. It can also produce enterotoxins that cause food poisoning, and immunity is insufficient to prevent reinfection. Question 2. True statements regarding streptococcus bacteria. 1. Streptococcus, the direction of division is constant, forming chains. 2. Streptococcus includes bacteria that form clear hemolytic zones on blood agar. 4. It is a gram-positive coccus. 5. Most streptococcus bacteria are anaerobic bacteria. 6. It includes bacteria that causes scarlet fever. Explanation. Streptococcus divides in a single plane, producing characteristics chain-like arrangements. Some species exhibit hemolysis on blood agar and include pathogens that cause scarlet fever. Question 3. True statements regarding Enterococcus faecalis. 1. It is an opportunistic infection. 2. Present in feces. 2. 3. Found in the human intestines. 4. A cause of urinary tract infection. Explanation. Enterococcus faecalis is part of the normal intestinal flora and is commonly found in feces, which is poo or shit. It can cause opportunistic infections such as urinary tract infections, especially in hospitalized patients. Question 4. Among diseases caused by S. aureus, which has a different mechanism of onset. 1. Food poisoning. Staphylococcal food poisoning results from ingestion of performed enterotoxin rather than bacterial infection. Symptoms occur rapidly without colonization or invasion of host tissues. Question 5. What are the gram staining characteristics and morphology of Staphylococcus? Answer. Gram-positive cocci. Explanation. Staphylococcus retains crystal violet stain due to its thick peptidoglycan cell wall. It's gram-positive, so it has thick cell wall. Microscopically, it appears as a grape-like clusters. Question 6. List the toxins produced by Staphylococcus aureus. Answer. Hemolytic toxins, exfoliative toxin, enterotoxin, and toxin shock syndrome toxin. Explanation. Staphylococcus aureus produces multiple exotoxins that damage host cells and tissues. These toxins are responsible for both localized infections and severe systemic diseases. Question 7. Which species belonging to Staphylococcus show no hemolysis on blood agar and forms white colonies and is therefore also called white Staphylococcus? Answer. Staphylococcus epidermidis. Explanation. S. epidermidis form white, non-hemolytic colonies on blood agar. It is a common skin commensal with low pathogenicity. Question 8. Which substance produced by Staphylococcus aureus binds to the Fc receptor of antibodies and is therefore used especially for isolating immunoglobulin G? Answer: Protein A. Protein A binds specifically to the Fc region of immunoglobulin G molecules. This property is widely used in immunological purification and diagnostic techniques. Question 9. Which enzyme causes coagulation by reacting with fibrinogen when S. aureus is mixed with serum? Answer: coagulase. Explanation: coagulase converts fibrinogen into fibrin, forming a protective cloth around bacteria. This helps Staphylococcus aureus evade host immune defenses. Question 10. What is the main causative organism of toxic shock syndrome? Answer: Staphylococcus aureus. Explanation: toxic shock syndrome is caused by superantigen toxins produced by S. aureus. These toxins trigger massive cytokine release leading to systemic shock. Question 11. In MRSA, what does the letter M stands for? Answer: methicillin. MRSA refers to Staphylococcus aureus strains resistant to methicillin. This resistance is due to altered penicillin binding proteins. Question 12. Which antibiotic is primarily selected as first-line therapy for MRSA, methicillin-resistant staphylococci, and enterococci? Answer: vancomycin. Explanation: vancomycin inhibits cellular synthesis in gram-positive bacteria. It is the drug of choice for MRSA and resistant enterococcal infections. Question 13. What extracellular enzymes are produced by Staphylococcus aureus? Answer: coagulase, lipase, hyaluronidase, DNase, and fibrinolysin. Explanation: these enzymes facilitate tissue invasion and bacterial spread. They significantly enhance the virulence of S. aureus. Question 14. What does the term coagulase negative staphylococci indicate as being negative? Answer: coagulase. Coagulase negative staphylococci do not clot plasma. This characteristic helps differentiate them from more pathogenic species. Question 15. Which surface substance of Streptococcus pyogenes bind to complement C3 and inhibit phagocytosis? Answer: M protein. M protein interferes with complement-mediated opsonization. This allows the bacterium to evade phagocytosis. Question 16. Which species belongs to the genus Streptococcus and possesses the group B antigen? Answer: S. agalactia. Group B streptococci are classified based on specific wall carbohydrate antigens. S. agalactia is a major cause of neonatal infections such as sepsis and meningitis. Question 17. Which toxin produced by S. pyogenes causes streptococcal toxin shock syndrome? Answer: erythrogenic toxin, also called pyroglutamic toxin. This toxin functions as a superantigen that strongly activates T lymphocytes. It induces massive cytokine release, leading to systemic shock. Question 18. What is the phenomenon called in which the capsule swells when pneumococci are treated with specific antiserum? Answer: quelling reactions. Explanation. The quelling reaction causes visible swelling of the pneumococcal capsule. It is used for identification and serotyping the Streptococcus pneumonia. Question 19. List the surface substances of S pyogens that allow it to evade innate immune system opsonization and phagocytosis. Answer. Hyaluronic acid capsule, M protein, M-like protein, C5a peptidase. Explanation. These factors inhibit complement activation and phagocyte recruitment. They play a central role in immune evasion and virulence. Question 20. Which bacterium was used to demonstrate that non-encapsulated R-form bacteria can transform into encapsulated S-form bacteria via DNA-mediated transformation? Answer. Pneumococcus, Streptococcus pneumonia. Explanation. Griffith's experiment demonstrated transformation from R-form to S-form bacteria. This experiment provided key evidence that DNA is the genetic material. Question 12. What are the two representative non-suppurative diseases caused by host immune responses following group A streptococcal infection? Answer. Acute glomerular nephritis, rheumatic fever. These conditions occur due to immunological cross-reactivity after infection. They are not caused by direct bacterial infection. Question 22. Explain the presence or absence of catalase and coagulase in Staphylococcus and Streptococcus. Answer. Staphylococcus is catalase positive and coagulase positive. However, Streptococcus is catalase negative. Explanation. Catalase and coagulase tests are essential for differentiating these different bacteria. These biochemical reactions are routinely used in clinical microbiology. Question 23. What are the two types of hemolysins produced by pyogenic streptococci? Answer. Streptolysin S. Streptolysin O. Explanation. Streptolysin O is oxygen labile, while streptolysin S is oxygen stable. Both toxins contribute to hemolysis and tissue damage. Question 24. Which infectious disease and causative organism are described below? Description. Strong immunity remains after infection. The tongue initially has a white coating that later peels, forming a strawberry tongue. Strawberry tongue. A pediatric infection characterized by high fever, rash, cloudy tongue, and leukocytosis. Answer. Scarlet fever. Streptococcus pyogenes. Explanation. Scarlet fever is caused by erythrogenic toxin producing group A streptococci. The toxin induces rash and characteristic strawberry tongue. Question 25. Briefly explain the mechanism of rheumatic fever following streptococcus pyogenes infections. Answer. Antibodies generated after bacterial infection cross-react with antigens in the heart and joints, triggering inflammation. Explanation. These conditions arises due to molecular mimicry between bacterial and host antigens. It represents an autoimmune-mediated complications. Question 26. Which of the following bacteria do not produce LPS and toxin? Answer. Staphylococcus, Clostridium tetani, Clostridium botulinum, Corynebacterium diphtheria. Explanation: endotoxin is a component of lipopolysaccharide found only in gram-negative bacteria. LPS endotoxin only found in gram-negative. This, yeah, yes. Question 27. When bacteria are cultured on blood agar and do not completely lyse red blood cells, producing a greenish zone around the colony, what is this type of hemolysis called? Alpha hemolysis. Explanation: alpha hemolysis represents partial destruction of red blood cells. It causes green discoloration due to oxidation of hemoglobin to methemoglobin. Question 28. What is a nosocomial infection? What is the opposing concept? Answer: nosocomial infection means hospital-acquired infection. The opposing concept is community-acquired infection. Question 29. Which bacteria are major causative agents of pediatric meningitis and have clinically used vaccines developed from capsular polysaccharides? Answer: Streptococcus pneumoniae and Haemophilus influenza type B. This bacteria possesses polysaccharide capsules that enhance virulence. Capsular polysaccharide-based vaccines are effective in preventing pediatric meningitis. Question 30. What are the scientific names of the following microorganisms? 1. Dysentery bacillus. Answer: Shigella dysentery, S. flexneri, S. bovidii, S. sonnae. 2. Cholera bacterium. Answer: Vibrio cholera. 3. Syphilis bacterium is related with Treponema pallidum. 4. Hay bacillus is related with Bacillus subtilis. 5. Epidemic typhus bacterium related with Rickettsia prowazekii. 6. Pneumococcus is related with Streptococcus pneumonia. 7. Typhoid fever bacterium is related with Salmonella typhi. These were the questions regarding gram-positive cocci. Thank you.